Greetings and welcome once again to another live and exciting session on the Professor Kareem Ainsley Encounter. Today we're going to be looking at the history of the anti-globulin test. Yes. The history of the anti-globulin test. If that is something that you're interested in knowing about, then meet us on the inside because class is in session. Before the anti-globulin test was developed, only the IgM antibodies were detected by direct testing methodology. So there was a gap. The introduction of the AHG anti-human globulin test permitted the detection of non-agglutinating IgG. Yes, IgG. That is the one that is able to pass the blood-brain barrier because of its small structure. And we know that the IgM is the large pentameric structure in most instances. So the IgG antibodies came about, and that then led to the discovery and the characterization of new blood group systems. All right. So in 1945, Coombs, Motrand, and Race describe the use of antiglobulin tests for the detection of non-agglutinin Rh antibodies in the serum. So yes, I know you have heard about the Coombs reagent. So now we know where we get the name Coombs from. And yes, you have Fisher race, that is the blood group classification. So we have an understanding and appreciation for these names and where they were derived. It was in 1946 that Coombs' co-workers described the use of the AHG in the detection of in vivo sensitization. This is very important. In vivo sensitization of the RBC of neonates, because we have in vivo sensitization and we have in vitro sensitization. So I want you to just have that in mind, that AHG was described in the 1946 to allow us to appreciate the in vivo sensitization of RBCs of neonates suffering from hemolytic disease of the fetus and newborn. We call it HDFN. And back in those days, they usually call it hemoblastosis fetalis. Yes. The test was originally called the Coombs test, but later we call it the DAT because we had a better application for it after a while. Although the Coombs and associates were instrumental in the introduction of antiglobulin testing to blood group serology, the principle of the test had been in fact described way before by Moresi, or Moreschi, in 1908. Yes, 1908. Murray's study used rabbit anti-goat serum to agglutinate rabbit RBC that were sensitized with low non-agglutinin doses of goat anti-rabbit RBC serum. So now we are seeing where we have one species with another species combining to give us what we now appreciate. The Coombs test involved injection of human serum into the rabbits to produce the anti-human serum. So that is how we have the anti-human serum coming about, because human serum is injected into non-human, usually the rabbits, to produce anti-human, because the non-humans produce antibodies that they don't have, which would be the human antibodies. So we have anti-human serum. So that's the simple essence of what is taking place. After absorption to remove the heterospecific antibodies and after dilution studies, guess what? We need this study to avoid prozone interference. The AHG serum still retains a sufficient amount of antibodies activity to permit cross linkage or linking of adjacent red blood cell sensitized with IgG antibody. The cross-link of the sensitized red blood cell by AHG produces heme agglutination. So now you have learned another technology, another technique that we practice in transfusion medicine or immunohematology. The cross-linkage of sensitized red blood cell by the AHG produce heme agglutination, which indicating that the red blood cell has been sensitized by the antibodies that have had reacted with the antigen present on the surface of that cell. So the red blood cell has antigens and antigen binding sites on the surface, and in the serum, or if you have plasma, you will have antibodies in circulation. Early, the AHG reagents were prepared using crude globulin fractions as immunogens. But in 1946, the Coombs-Montre demonstrated that the antibody activity that detected the Rh antibody was associated with anti-gamma globulin, the fraction. So yes, we have now a new discovery. You see, we have discovery after discovery. It is one research building on another research, and that is why scientific studies are very important and crucial, especially in the area of transfusion medicine and laboratory medicine as a whole. And for that, we have to continually be doing our continuing education. We have to be continually doing research, trying to detect and decipher new techniques in order to ensure that the process is smooth and streamlined. So let us wrap up this history so you can get into the meat of the matter as class is in session. So in 1951, Dice presented the first evidence that there might be other antibody activities present that influence the final reaction. He observed that different reaction patterns were observed or obtained when dilution of AHG were used to test cells sensitized with a warm IgG 37 degrees as compared with a cold IgM at less than 22 degrees. All right, so we're seeing cold reacting or room temperature reacting and warm reacting. So the IgG are the ones that normally react at the 37 degrees, and these are clinically significant, while the IgM, they have significance, but most of the time they are not clinically significant. But when they become clinically significant, it is very important for us to identify them immediately. It was in 1957 that Dice and his co-worker discovered and published data showing that the reactivity of the AHG to cell sensitization with warm antibody resulted from anti-gamma globulin activities, whereas the non-gamma globulin activities were responsible for activities of cells sensitized by cold antibodies. So the non-agglutinable or the non-gamma globulin complements were shown to be beta globulin, all right, and has a specificity for complements. So when we talk about complement, and I want two things to come to your mind: C3 and C4, all right, which will be revealed in later studies. All right, the anti-globulin test can be used to detect red blood cell sensitization with IgG alloantibody or IgG autoantibodies and complement components. So sensitization can occur either in vivo, all right, that is within, or in vitro, that is without or outside of. The use of the AHG to detect the in vitro sensitization of RBC is a two-stage technique referred to as the indirect anti-globulin test, or the IAT, while the in vivo inside sensitization is detected by a one-stage procedure called the DAT, which is the direct antiglobulin test. The IAT and the DAT still remains the most common procedures performed in blood bank group serology, and we're going to get into this technology some more so that we can have an appreciation as to how it works and the significance. So this is just a brief summarization of the history of how the antiglobulin tests came about. This is here food for thought. At this time, I allow you to digest the information as we take a break in this class session. See you after the break.